So over the last few years, we've been performing genomic characterization of brain metastases, and we've done a large-scale effort, but today I'll present just the initial results of our first data set that we published a couple weeks ago, actually in Cancer Discovery. And I'll talk about the implications for cancer and precision medicine in brain metastases. So brain metastases are an unmet clinical need, as you all know. So autopsies have reported up to 25% of cancer patients will develop brain metastases. Most are derived from lung, breast, and melanoma. And the incidence is rising as systemic therapies are getting better and patients are living longer. So patients will often develop brain metastases or progressive brain metastases in the setting of extracranial disease that is adequately controlled with existing chemotherapies or targeted therapies. Historically, it's been unclear if this clinical divergence is due to inadequate systemic therapeutic penetration of the blood-brain barrier versus different genetic drivers in the brain metastases. And we have a limited understanding of how brain metastases genetically evolve from their primary tumor. So the clonal theory of cancer evolution states that starting from a single normal cell, across molecular time, multiple rounds of mutation and expansion occur. Eventually, individual clones break off and give rise to metastases, which then undergo further evolution. So the historical model of metastasis formation is that metastases are descended from a single cancer clone. Another possibility is that multiple subclones contribute to a single metastasis. And more recently, data from mouse models has supported a more complex process, whereby cells derived from distant mets can then reseed their primary tumor. So in the last few years, there have been a handful of deep sequencing studies that have explored the genomic differences between different metastatic sites within the same patient to understand the evolution of the metastatic process. So the largest massively parallel study in brain metastases was an N of 1. And all of these studies of small numbers of patients have confirmed clonal evolution, organ-specific branches, and significant intratumoral heterogeneity, specifically within the primary tumor. And a recent study looking at multiple metastases, not brain metastases, in 10 patients with prostate cancer showed metastasis to metastasis seeding as well as self-seeding. And prior data from targeted sequencing array CGH suggests activation of the PI3 kinase pathway in brain metastases. So the objectives of our study were to 1, elucidate the evolutionary patterns leading to brain metastases, 2, identify whether brain metastases harbor clinically significant genetic differences compared to their primary tumors. 3, determine the extent to which clinically significant mutations are shared among regionally, anatomically, and temporally distinct brain metastasis sites. And 4, to determine whether lymph nodes or extracranial metastases are genetically similar to brain metastases and might serve as their proxy for clinical decision-making. So we performed whole-exome sequencing of 104 brain metastases matched with primary normal tissue. And this included 15 with additional extracranial sites or temporally, regionally, and anatomically separated brain metastases. So this has been a tremendous multidisciplinary effort, both within our institutions in Boston and internationally. And why did we need such a collaborative effort? Well, brain metastases were not routinely banked. The primary tumors were resected 10 or 15 years ago. Also, DNA extracted from older blocks was of poor quality, and consents required for whole-exome sequencing continue to be quite rigorous, and older samples may not have been adequately consented. So this is a summary of our cohort. The majority of our patients are derived from lung and breast cancers. The median time to diagnosis of the brain metastasis from the primary tumor was 2 years, but there was significant variability in our patients. And the median number of treatment regimens between the primary and the metastasis was an N of 1, but again, significant variability. So when carrying out these large-scale genomic sequencing studies, the first step is to determine the genome alterations that are unique to the patient's cancer, so shown on the left here. And then we try to understand which alterations are recurrent and statistically significant in the population of samples that we're looking at. And there's different algorithms that are in place in different institutions to do that. So these are just the list of algorithms we use at the Broad Institute to determine these alterations. So if we use those same algorithms that are used in other large-scale sequencing studies of primary cancers to look at what's recurrently and significantly mutated across the group of our primary tumors in our brain metastases, so when we're looking at the most common mutations across the metastases and the primary tumors analyzed separately, we find P53 and KRAS as the top hits. So each column here represents one sample. The histogram here is the mutation rate of each of our samples. So you can see P53 and KRAS are the most common mutations. This doesn't tell us much about the genes important in metastases, nor does it tell us much about the clonal path from the primary tumor to the metastases. These are the copy number profiles across our primary and metastatic samples. So blue is copy number loss and red is copy number gain. You can see that these are all very complex. So how do we use all of these data to elucidate the evolutionary patterns in each brain metastasis sample? So before I describe the analysis of our sequencing data, let's think about what's actually in a cancer tissue sample. So the somatic evolution leading up to cancer diagnosis and sample collection results in a heterogeneous mixture of related cancer clones as well as normal cells. And the number varies substantially from one sample to another. And when we are trying to understand how the fraction of mutations in each matched metastasis and primary tumor relate, it's particularly important to understand and correct for this variability in purity and ploidy. So how do we do this? How do we deconvolve cancer tissue samples? So DNA sequencing quantifies the relative abundance of variants or alterations present in the cancer tissue cells. Our collaborators, namely Scott Carter and Gadi Getz's group at the Broad Institute, developed a computational method called absolute, which uses Bayesian statistical models to correct for purity and ploidy and to estimate a cancer cell fraction for each mutation. So cancer cell fraction is the percent of cancer cells that have a particular mutation. So mutations that have a cancer cell fraction of 1 are present in 100% of the cells. They're clonal in that biopsy, as represented by the green dot. Mutations that have a cancer cell fraction less than 1 are subclonal. They affect a portion of cancer cells. So the pink dot here is subclonal. It's present in half of the cancer cells sampled. Therefore, its CCF value is 0.5. Subclonal mutations occur later in evolution. And when we analyze a paired primary tumor and brain metastasis, we plot the cancer cell fraction values for each mutation, shown in the left panel here. Primary on the x-axis, metastasis on the y-axis. So clusters of mutations with similar cancer cell fractions are shown. And the numbers represent the size of each cluster, or the number of mutations in each cluster. So these represent different clonal or subclonal populations within each sample. We then developed a computational method called phylogic, which automates the inference of the phylogeny in each cancer case from this cancer cell fraction plot, so shown on the right here. So now we draw a quantitative phylogenetic tree by making the branch lengths proportional to the number of mutations in each cluster. And then we label branches with known oncogenic drivers or cancer drivers. So these data here are from a real example of an esophageal carcinoma that metastasized to the brain. So let me walk you through one of these plots. So in the top right corner here, these mutations are present in 100% of the cancer cells sampled from the brain metastasis in the primary tumor. So they're thus clonal in both biopsies. So this cluster of 261 mutations, it's the most recent common ancestor of the primary tumor and the metastasis. Then there's continued evolution within the brain metastasis, as depicted here by the clonal cluster of 45 mutations and the subclonal cluster of 21 mutations that are not present in the primary tumor biopsy. And subclonal mutations in the brain metastasis, so they're novel mutations in the brain metastasis that happened after the brain metastasis occurred. And then similarly, we see further evolution within the primary tumor with a new subclonal cluster of 18 mutations and a clonal cluster of 14 mutations. So this depicts a single clone origin of metastasis with branched evolution. So in this relationship, the brain metastasis and the biopsy of the primary tumor, they share a common ancestor, but then there's continued evolution in the primary tumor reflected by these clonal mutations that are in the primary biopsy that are absent from the brain metastasis. So here, the primary and the brain metastasis are related or evolutionarily siblings. So across all our related samples and phylogenetic trees, we find that the data is consistent with this same single clone relationship with branched evolution, where we detect the common ancestor, but then there's continued evolution within the primary and the brain metastasis. Interestingly, in 4 of our cases, there were no shared mutations, so mutations present here, between the primary tumor and the brain metastasis, suggesting that these were clonally unrelated samples. So interestingly, 3 of these arose in the lungs of smokers that had multiple histologically distinct primary tumors that were diagnosed clinically. And an additional case was derived from a BRCA carrier that had a second primary breast cancer detected in the contralateral breast. So this suggests that these metastases arose from the unmatched primary tumors that we did not sample. And notably, our data was not consistent with self-seeding, whereby one would see mutations reseeding the primary tumor, depicted by the green and black ovals here. And nor was our data consistent with the polyclone or multiclonal origin of metastasis, whereby one would see mutations depicted by the green circles here to suggest that multiple clones contributed to macroscopic metastases. So mutation clusters that are present subclonally in both samples can only occur as the result of multiple seeding events by 2 or more genotypically distinct cells. So as a caveat to all of this, these are all clinical biopsies that sampled only very small regions of the primary tumor and the brain metastasis. So this may have contributed to this predominance of our branch sibling relationship that we saw across all our samples. And notably, we also did not detect a linear pattern of spread, whereby we did not find the subclone within the primary tumor, which was the direct subclone that seeded the brain metastasis. In all cases, we detected a branch sibling relationship. So the primary tumor and the brain metastasis shared a common ancestor, but there was continued evolution. So how is all of this relevant clinically? So do brain metastases harbor clinically actionable mutations that are absent in their primary tumors? So to answer this question, we annotated the phylogenetic branches with mutations in 121 driver genes from the target database of clinically actionable genes. So these are known clinically actionable genes that alter patient management in oncology, many in the context of genomically guided clinical trials in oncology. And we were rigorous with the criteria, so for tumor suppressors, we required biallelic inactivation, and for oncogenes, amplifications or hotspot mutations. And we did indeed find that brain metastases are genetically distinct from their primary tumor biopsies. So this is a representative example of a patient with a primary renal cell carcinoma that was diagnosed synchronously with the brain metastasis. So you can see that mTOR and VHL are in the common ancestor of the primary tumor and the brain metastasis, but there's a clear driver mutation occurring in PIK3CA, this is a hotspot mutation, that's only in the brain metastasis. And we performed deep sequencing of the primary tumor and did not detect that mutation in the primary tumor. This is a representative phylogenetic tree of a breast cancer sample in a patient with a HER2-positive breast cancer who developed a brain metastasis while being treated with trastuzumab therapy. So here, we detected a subclonal EGFR mutation exclusive to the brain metastasis. And although this particular mutation is common in lung cancers and predicts sensitivity to EGFR inhibitors, one proposed mechanism of resistance in anti-HER2 therapy in breast cancer is activation of EGFR. So this is consistent with a resistant subclone that was selected for during trastuzumab therapy. And we confirmed the absence of the EGFR mutation in the primary tumor with deeper sequencing. This is another representative phylogenetic tree of a renal cell carcinoma. So here the patient developed a brain metastasis while on bevacizumab 3 years after initial diagnosis of a primary renal cell carcinoma. So here we see a biallelic inactivating event in PTEN exclusive to the brain metastasis. In this example, interestingly, we also found evidence of convergent evolution. So PBRM1 is commonly mutated in renal cell carcinoma. So here we see a different PBRM1 frameshift mutation in each of the primary and metastasis, and these are likely loss of function events. This is a representative phylogenetic tree of a patient with an ovarian carcinoma who developed a solitary brain metastasis 5 years after being in complete remission from a primary ovarian carcinoma. And here we detected a HER2 amplification only in the brain metastasis sample that was not detected in the clinically sampled primary tumor. And this was validated with IHC data shown in the left here. So here's an example of a HER2 amplified breast cancer that had stable extracranial disease and then developed a brain metastasis after 3 years of trastuzumab therapy. The brain metastasis and the primary tumor shared an amplification in ERB2 and a homozygous deletion of p53. However, the primary tumor had an additional MYC amplification, shown here, that was not detected in the brain metastasis sample, and the brain metastasis harbored a homozygous missense mutation in BRCA2 that was not detected in the primary tumor sample. And deep sequencing, again, of the primary tumor sample failed to detect the BRCA2 mutation. So when we look across all our samples, clinically actionable alterations are present on any branch of the phylogenetic tree. So here is a histogram of all clinically actionable alterations detected that occur in each case. And colors represent what branch they occurred on. So gray are shared by the primary and the brain metastasis, red exclusive to the brain metastasis, blue exclusive to the primary tumor. Importantly, 53% of cases have a clinically actionable alteration in the brain metastasis branch that was not detected in the primary site. And we group the different genetic alterations into what inhibitors they may predict sensitivity to. And here I just show a very small set of clinically actionable genetic alterations, and each column represents a different case. And different colored boxes, again, represent what branch they occurred on. And you can see that clinically relevant mutations occur on all phylogenetic branches of these cancers, including in the brain metastasis-specific branches. The most common alterations were those predicting sensitivity to CDK inhibitors. So here's a list of genes that were altered. Each column, again, represents one sample. Each color represents what branch of the phylogenetic tree they occurred on. And different symbols represent the types of mutations that were detected. So 51% of cases had alterations associated with sensitivity to CDK inhibitors. And these alterations included CDK4/6 amplifications, as well as loss of CDKN2A, along with a number of other alterations. Mutations affecting the PI3 kinase pathway were also frequent. So actionable alterations in these genes occurred frequently in breast cancers, lung cancers, and renal cell cancers. And alterations included PIK3CA mutations and PIK3R1 mutations, as well as PTEN loss. We also found mutations that predict sensitivity to HER2 and EGFR inhibitors, as one would expect. So 13 breast cancers had amplifications in ERBB2, and all of which were shared by the brain metastasis in the primary tumor. In one case, we detected an additional activating ERBB2 missense mutation only in the brain metastasis sample, in addition to the shared ERBB2 amplification shown here. And notably, 2 patients with lung cancer and a third with ovarian cancer, the one that I mentioned earlier, had ERBB2 amplifications only detected in the brain metastasis samples. And 2 patients with HER2 amplified breast cancer had EGFR alterations, again, detected only in the brain metastasis sample. So the MAP kinase pathway inhibitor family includes agents that inhibit BRAF and MEK, such as vemurafenib, dabrafenib, and trametinib. Activating mutations in KRAS were the most frequent alteration in this group, and the majority were shared, as shown by the gray bars here. So genetic divergence between primary and metastatic samples poses a major challenge to precision medicine in oncology. What about regional heterogeneity within the brain itself? How representative of bulk CNS metastatic disease is a single brain metastasis sample? To answer this question, we sequenced regionally, anatomically, and temporally distinct areas of brain metastases. So here's a representative case of a patient with a primary HER2 breast cancer that developed a solitary brain metastasis 7 years after initial diagnosis. Here we sequenced 4 different regions of a cerebellar mass. So shown here, and as you can see here, all 4 regions were more closely related to each other than either was to the primary sample. So the length of line here is proportional, to remind you, to the mutation rate. So all 4 regions of the brain metastasis shared actionable amplifications in CCNE1 and EGFR, shown here, that were not present in the primary tumor. Notably, the ERBB2 amplification that we show here is present in both the primary and the brain metastasis in the shared clone. So temporally distinct brain metastases also shared clinically informative drivers. So this is in a patient with a large cell neuroendocrine lung cancer, and here we sequence resections of the brain metastases before and after whole brain radiation. And we found that each sample before and after whole brain radiation shared the same MYC amplification. And that was not detected in the primary tumor sample. And again, the regions were more closely related to each other than either was to the primary sample. So this is another example of a patient with a triple negative breast cancer who underwent a resection for a symptomatic cerebellar metastasis and 2 months later had rapid local recurrence, necessitating re-resection. And the primary tumor and the brain metastasis shared alterations in P53, P10, and MYC, shown here. And the primary tumor harbored an additional MCL1 amplification not detected in the brain metastasis samples. But we also identified an additional mutation in EZH2 in both brain metastasis samples, shown here, but failed to detect this mutation in the primary tumor sample. So interestingly, clinically actionable mutations were shared even in metastases in anatomically distinct areas of the brain. So here's a representative example of a patient with a salivary gland ductal carcinoma who had a symptomatic cerebellar tumor resected pre-whole brain radiation and a parietal metastasis resected post-whole brain radiation. And here we sequenced 2 regions of the cerebellar metastasis as well as the parietal metastasis, and we found that both regions of the cerebellar metastasis and the parietal metastasis were more related to each other than either was to the primary tumor. And both the cerebellar and parietal metastases shared the same amplifications in MET, CDK6, cyclin E1, AKT2, and MYC. And those were not detected in the primary tumor biopsy, so shown here. So metastases within the central nervous system are relatively homogeneous. So given that central nervous system disease may be difficult to access clinically in some cases, we then evaluated the extent to which involved regional lymph nodes and distal extracranial sites, as well as other sites of the primary tumor, recapitulated genetic vulnerabilities present in the CNS lesions. So in this example, we sequenced the primary ovarian cancer and an involved regional lymph node, as well as the brain metastasis. Although genomically, the lymph node was more related to the brain metastasis here, the brain metastasis had an additional aurora kinase A amplification that was not detected in the primary biopsy or the lymph node. In another case, we sequenced a triple-negative breast cancer and an involved regional lymph node. And here, the primary shared a common ancestor with the brain metastasis that was not shared by the regional lymph node, shown here. And here, the primary had an additional EGFR amplification that was not present in the brain metastasis or the regional lymph node. And just as lymph nodes were not reliable surrogates for clinically informative alterations in the brain metastases, distal lesions were also genetically divergent. So in this example, we sequenced a primary colorectal cancer, a lung metastasis, and a brain metastasis. And here you see that the brain metastasis had more alterations that were unique to the brain metastasis than was shared by all samples, so as depicted by the long red branch here. And here we also observed an amplification in MYC in the brain metastasis that was not present in the primary tumor. And finally, we sampled 2 regions of a primary lung cancer, one before and one after 2 cycles of neoadjuvant chemotherapy and chest radiation, in addition to sequencing the brain metastasis, which was diagnosed 5 months later in the absence of any extracranial disease. And here the brain metastasis harbored, again, mutations that were not present in either primary sample. So brain metastases are genetically divergent from primary tumors and other extracranial sites. So we demonstrate divergent or branched evolution across all our cases. How is this important clinically? So if one were to exclusively sample the primary tumor or extracranial sites for selection of the appropriate targeted therapy, one may miss potentially actionable alterations within the brain itself. So our data show that clinically actionable variants occur in the brain metastasis branch 53% of the time. And that presents a significant challenge to the application of precision medicine to brain metastases. And furthermore, patients will often develop progressive brain metastases in the setting of their extracranial disease that's controlled with existing therapies. Here, the observations suggest that additional oncogenic drivers or alterations may be present in brain metastases, and this may contribute to the divergence of therapeutic response that we see in brain metastases and other extracranial sites. So in conclusion, every brain metastasis displayed branched evolution. Brain metastases harbored distinct clinically actionable genetic alterations compared to their primary tumors. Different brain metastasis regions are relatively homogeneous, and extracranial metastases are not a reliable surrogate for brain metastases. And I'd like to thank a number of individuals and institutions who've contributed to this work. So these efforts obviously take an army of individuals to help, and I'd like to thank namely Scott Carter, Tracy Batchelor, David Lewis, and Gadi Getz, as well as all the patients who very generously donated their tissue samples for this study. And I'll take questions.
Two very brief questions. Did you actually act upon any of the actionable mutations? And the second one, that in the esophageal and lung cancer cases, I'm assuming that they receive platinum-based therapy. So when you see those extra mutations in the brain, do they look like platinum-induced mutations? They have a quite, I'm sure you know, specific mutational pattern. So I was just wondering whether that is consistent or not.
So good question. In terms of acting on mutations, we did have one patient on this in this data set that had gotten a PI3 kinase inhibitor and had a PI3 kinase mutation in the brain and actually did very well and didn't progress intracranially. But that was just a vignette. We're now moving forward with the genomically guided brain metastasis trial. So we're starting that at MGH within the next couple of months. And so what we do need to show now is that by treating what we see in the brain, we need to show it prospectively that we can improve survival outcomes. So we're starting this trial. We've also presented this to the Alliance NCI Cooperative Group, and there's a lot of interest to make this a multi-center trial, essentially like a matched trial, but one for brain metastases patients. So that's the answer to your first question. The second question, great question. So we're actually looking at the lung cohort a little bit more carefully now, so to actually see if we see the signature of chemotherapy. So in this particular cohort, we didn't, but now we have about 75 lung cancer brain mets in a separate cohort, so we're going to be looking at different signatures and correlating with treatment response. We didn't have, so in this particular cohort, because these tissue samples were collected over 15 years, or these were very old samples also, many of them had a lot of heterogeneity in their treatment. So, and many of these were either synchronous, some of these had gotten targeted therapy. So now that we have a larger cohort, we'll be able to look more into how treatment correlates with what we saw in the genetic mutations. But great questions. Yes?
So two brief questions. So the first is, to what extent are the brain metastases heterogeneous themselves? It was hard to kind of get that because that also will confound precision therapy. And the second question is, how do you explain the difference between your results and the previous studies, especially regarding self-seeding and other models of metastasis, polyclonal metastasis from the primary, for example?
So the answer to your first question, so the brain metastases themselves, so anatomically and temporally distinct brain metastases, were actually relatively homogeneous. So especially compared to the primary tumor. So we actually found that across the entire, again, this cohort only had a small number of anatomically and temporally distinct brain metastases, but the ones that we did look at, they were relatively genetically homogeneous, which is actually reassuring that a single brain metastasis biopsy or tissue from a clinically sampled brain metastasis may be sufficient to identify most clinically informative drivers within the brain itself. So that's the answer to your first question. The second question, so in this case, we only sampled small regions of the primary tumor as well as small regions of the brain metastases. We had more tissue for the brain metastases more so than the primary tumor. So that might explain why we didn't see evidence of self-seeding or the multiclonal seeding. So we might have been limited because we had small numbers, only a small sample of these tissue samples. However, we didn't see across the entire all 100 brain metastases, we didn't see any evidence of the self-seeding. So it might be a blood-brain barrier, issue where we might just not be seeing self-seeding as much in brain metastases.
Could it be a difference in the algorithms used for analysis? So if you go back and look at the previous data that were published with your algorithms, do you get a different answer?
So we're actually gonna look into that. We have some collaborators who are interested in looking at that, but that's a great question.
Tito? Tito, you probably don't have the answer to this yet, but I guess you could probably get it eventually. I'm surprised that you say that different brain metastases are so similar, because that's not what's thought to occur for other sites of metastasis. So you should have eventually enough data that you could look to see whether it's more true that in the brain there's more homogeneity than there is elsewhere.
Yeah, so we actually have a very robust autopsy program going now. So where we're going to actually where we're sampling multiple distal mets and multiple regions of distal mets, as well as multiple tumors, multiple primary regional sites, and then also looking at brain metastases. But yeah, exactly. So we'll have more data. So this was only a small number, but we'll have more data to actually conclusively say that. Great question.
It's hard to biopsy brain tumors in patients, and particularly when there are multiple sites. What kind of information can you get from circulating tumor cells or circulating DNA?
So great question. And I'm not advocating for brain biopsies in every patient yet. I think we first need to show that by targeting what we see in the brain from tissue that's already clinically available, that will improve patient outcomes. To answer the question, so we're actually looking now into CTCs and circulating DNA to see if we can answer that question. I think the challenge is going to be there is knowing whether what we see in the CTC recapitulates what we see in the brain versus what's in distal sites. So I think it's going to be a huge challenge, but we are going to look into that. But I think even with circulating DNA, it's going to be hard to know if what we see circulating in the blood can serve as a surrogate for what's actually in the brain. So I think it's going to be a challenge.